I want to make sure we're getting to fill finish, certainly, in before we a couple more topics before we officially turn over to our audience Q&A. Um, Specifically with regard to fill finish, I want to hear perhaps from each of you, the analytical methods that you're employing to detect and quantify trace levels of cryoprotectants and perhaps other excipients in the final cell therapy product. I would offer it up to all three of you, but perhaps Pratik, can you start us off?
Yeah, so I mean, I think it really comes down to how you assess risk. And so oftentimes what people will do is they'll start by looking at the concentrations of the formulations that are, or the components that are in the formulations that work their way from, let's say, drug product all the way to what actually is administered to a patient. And you can do a paper-based exercise to be able to look at, based on the number of washes and concentrations and other things, what drives what we think would be a worst case calculation for a residual amount in what a patient might see. And then you can work backwards from that and look at literature, and you can work with outside parties to help you determine if that is anywhere near what might be deemed unsafe for a patient. And if you're anywhere near it, and different people will use different metrics, maybe you're a tenfold higher or lower, or whatever that number is, but you have some kind of risk-based tolerance there. And anything that's maybe close to what might be an exposure limit, you'd work backwards and say, well, how do we develop an assay that would then help you understand the actual residuals? So you can start taking your paper-based exercise and translating it into an actual assay that you would run to determine starting concentration, end concentration, to be able to quantify it. You don't have to do that for every component, especially as you're getting into a first-in-human type trial, but it is ideal that you do it for certain categories of components that you might identify some risk associated with. So that's maybe a general approach that you might start, and that would help you.